Hello, everyone. I want to welcome to the webinar here today. We're going to give it about another minute or two while we wait for some more people to log on, and then we'll get started. Hello, everyone, again, and welcome to today's webinar. My name is Dusty Howe, and I am the Marketing Manager at Sexton Biotechnologies. Today, we'll be discussing how to rethink the security and supply of blood-derived ancillary materials in the era of novel viral outbreaks. This webinar is co-hosted by Sexton Biotechnologies and Hitachi Chemical Advanced Therapeutic Solutions. Sexton Biotechnologies is focused on the development and sales of bioproduction tools for cell and gene therapy. Their products and media enable flexible automation and scaling of cell manufacturing processes to increase the probability of positive clinical outcomes and reduce time to market, failure points, and labor costs. Hitachi Chemical provides cell and gene therapy contract development and manufacturing organization services at current good manufacturing practice standards, including clinical manufacturing, commercial manufacturing, and manufacturing development. The global footprint of this business is over 200,000 square feet and includes operations in North America under the name Hitachi Chemical Advanced Therapeutic Solutions, in Europe is Apseth Biopharma, and in Japan. We have three presenters on today's webinar. These include Alex Klarer, Senior Biomedical Engineer at Hitachi Chemical Advanced Therapeutics. Alex will be speaking on the supply chain challenges posed by human-derived ancillary materials and how the current public health emergency may exacerbate them. Our second speaker, Sean Werner, is a president at Sexton Biotechnologies. He will discuss how Sexton approached adding to the safety and security of their pooled human platelet lysate to meet ever-evolving regulatory requirements and protect against unexpected novel safety risks. Our final speaker, Norihiro Watanabe, instructor at Center for Cell and Gene Therapy, Baylor College of Medicine, has been working on CAR T cell treatment for both hematological malignancies and solid tumors. He will discuss how Sexton's pathogen-reduced human platelet lysate positively impacted the manufacture and function of CAR T cells. Finally, before we get started, we wanted to make you aware that you can submit questions during the presentation by using the Q&A function provided at the bottom of your screen. We'll do our best to get to as many of these during the final 15 minutes of the webinar. And now, without further ado, we'll begin the presentation with Alex. Take it away, Alex.
Thank you, Dusty. We can go to the next slide here. At Hitachi Chemical Advanced Therapeutic Solutions, we take this concept of an ideal cell therapy manufacturing campaign, which is your needle-to-needle process executed over time, and break it into these high-level measures for achieving a high functional manufacturing campaign. And as we engage with you, we add a little more specificity and expand each of these ideas into your exact specifications based on your critical quality attributes. But having an ideal target allows us to progress your process through each clinical stage toward that ideal point. And today we're going to drill into the idea of your robust and secure supply chain. If you want to go to the next slide, Dusty, with a particular focus on human-derived ancillary materials, because those present a unique challenge for cell therapy manufacturers, especially as you scale up and you start to require a larger volume of your reagents, and you put additional stress on what can be already fairly bottlenecked supply chains. Go to the next slide. We want to work with our clients to build these supply chains. So looking at that conceptually, a robust supply chain is one where all your reagents and materials can be sourced at cGMP quality from multiple sites. And we understand that there's going to be a transition period from this pre-IND clinical translational phase in through late phase and commercial manufacturing. But it's about laying that foundation with your reagent choices, especially with ones that you are identified as higher risk, and ensuring that if it's viable to not go straight toward a GMP quality for a phase 1 clinical trial, which is acceptable per the regulatory bodies, that there is a framework for moving that forward without high burden on you to show comparability or to bridge between reagents. And then you also want to look at your supplier and ensure that, one, they have shown sustainability as a company, and that they can overcome challenges like we're seeing today. If you're working with a small company that doesn't have a support structure, you're going to have difficulty using them through economic downturns and through unique conditions like we're experiencing today. And so you want to make sure that you're developing that manufacturing process to meet late stage supply chain requirements early, and select products that, if they can't be transitioned into a GMP quality, they have analogs that are easily exchangeable later in your clinical progression. Next slide. And that's all part of the quality by design approach. It's not all about realizing that 100% scalability and sustainability in phase 1 and phase 2, but it's laying the foundation, having that planning and development in place that allows you to take a phased approach to the development of your manufacturing process. And in line with that, the supply chain is a supporting function for all of these different quality attributes for your manufacturing process, and that needs to be defined early. Next slide. And then if we drill into blood products, they present particularly difficult supply issues where your volunteer donors must meet eligibility requirements, and those can be fairly stringent, especially when you're talking about human AB serum, for instance. You're sourcing that from male AB positive blood donors, right? And the more therapy developers who are using that reagent, the more restricted your supply chain becomes, because that's not a flexible source. It's not easy to increase the number of male AB positive people in a certain population. So you're working with an inflexible supply, and every individual donor biology is going to lead to variation in that product efficacy. And so different suppliers have different strategies for dealing with that variability, but a lot of times what a therapy developer will do is just buy an entire lot of a human-derived material in order to account for that. And what you're really doing then is using money, using capital to make up for the fact that this reagent does not have sustainability or scalability to that meets your requirements. And so you're making large upfront purchases in order to account for that. And lastly, blood donations have the possibility to transfer pathogens from donor to patient. And a lot of those pathogens are well defined and captured in current analytical procedures. But we had an instance 2 years ago where Zika made it into the US, and that was a test that these collection facilities all of a sudden needed to account for and to adjust their release procedures to test for the Zika, find maybe third-party testing facilities to perform that for them. And we're seeing today that there's a high risk for unknown pathogens to rapidly spread through today's society. And so that brings us to this concept, next slide, Dusty, thank you, of a black swan event. And this is something that was originally talked about in the investment community, but I think it really applies across the spectrum, where it's an unpredictable event that is beyond what is normally expected, and it has potentially severe consequences. They're also characterized by extreme rarity and severe impact. So what this is saying is we're not ready for the cascade of effects from this event, and we haven't seen something like this in the past in order to very accurately predict what the responses are going to be. And we're living through this right now. And to not take away from the short-term measures that we need to take to bridge us through this health emergency, but there will be long-standing impacts of this pandemic, and we need to consider what those are going to be for the cell and gene therapy industry. And we're considering its impact, particularly to human-derived ancillary materials, as one of those significant changes that we're going to need to adapt to. Next slide. So this ongoing health emergency has already impacted how we as a society and how our regulatory officials view public health policies. And we're seeing a large change in how the FDA is dealing with at-home testing and quick release and quick review of COVID-19 related measures. But we could also see an impact to eligibility requirements for donors. So how are we going to pre-screen? How are these suppliers going to pre-screen their donors? Is that going to further restrict the donor pool? We're already worried about the number of people who can provide blood, who can provide plasma and platelets. So how is this going to restrict that pool further? And then we also have a potential to see changes to where they're able to donate and the procedures by which they donate. We're looking at long-term social distancing policies. So is that going to change the throughput for these clinics? Are they only going to be able to have a certain number of patients per square footage that is severely restricted per to what they're able to service today? So even if the donor pool is able to meet the demand, are the suppliers going to be able to have the throughput that meets the demand? And we touched a little bit on, touched a little bit on testing before too. There's a potential for an impact in testing requirements, particularly on the front end to screen patients to make sure you're not bringing in any currently infected people to the clinic, the testing or the collection facility, and some increased costs associated that are passed down to the end user. And then are there going to be changes to how the regulatory bodies react to new pathogens? We saw that happen once where Zika was added to the testing protocol, and that only happened once Zika reached mainland US. are we going to start to see regulatory bodies be more cautious and start to add pathogens earlier on in the cycle as soon as they emerge, if there's a potential for them to be translated through a blood product? And then the developers, the manufacturers, as end users, need to adopt flexible procedures to accommodate changes to the regulatory environment. So both on being able to change suppliers easily if the supplier isn't able to meet these new requirements, and have a quality system that can accept variable C of A's if the C of A needs to change, if the accessioning procedure needs to change. Next slide, let's see. And so to make that cascade a little bit more specific, for allogeneic therapies, they may face a reduction in their identified donor pool, which I know a lot of allogeneic therapies have a very restricted subset of people they want to collect from. And those donors might opt to provide material to the current COVID-19 effort in the short term, or they'll be unable to donate because of exposure to the pathogen. And in the long term, we may see a shift in willingness to expose themselves to clinical site and to engage in elective procedures for fear of contracting some illness. We see a change to the mores of our society. And then looking at the human-derived media supplements, we already know these are a supply bottleneck, and we expect that bottleneck to only become more acute if we can't address some of the risks that are being elevated in today's environment. And so we're going to see a pathogenic risk from these reagents increase in an environment where novel pathogens are transmitting through the population easily, and our testing protocols are struggling to keep up. And therapy developers need to prepare for increased regulatory scrutiny of human sera and blood-derived compounds such as HSA and platelet lysate, and potentially look at moving toward reagents that are pathogen reduced or recombinant to account for that. The next slide. And then just to bring these into some questions you can ask your teams, you can ask your suppliers. Can your supply chain withstand a reduction in viable human donors, or reduction in the supply of these ancillary materials? And do your suppliers have strong enough quality systems to accommodate a dynamic regulatory environment, or are they struggling just to bridge their current quality system into a GMP quality system? And then how are human-derived reagents assured against currently unknown pathogens? And so with that, I'm happy to pass it off to Sean or to Dusty.
Yeah, perfect. Thank you, Alex. Really appreciate it. We'll continue now with Sean Werner from Sexton Biotechnologies.
Thank you, Dusty. Thank you, Alex. So we're going to pivot a little bit now to Sexton and talk a little bit about how we've approached these same kind of security and safety concerns leading up to this time, and how we're accommodating it currently. So just briefly, a little bit about Sexton. We're a relatively new company that spun out from Cook Regentech, bringing with us tools intended to be used in the cell and gene therapy space. Our goal is to really continue to develop tools and media products that help improve the performance of our customers' final products, increasing probability of success in clinical outcomes, and also to bring flexibility into manufacturing automation, integrating different components of the unit operations. Obviously, everybody is well aware of the complexity in cell and gene therapy manufacturing. Not only do you have to be able to show the science works in clinical effectiveness and develop your robust GMP processes, but the entire range of decisions that you have to go through to figure out even how you're going to manufacture your product is extremely broad, not standardized, and there's a lot of decisions that you have to make. And some of those decisions come back to your ancillary materials. So clearly the ancillary materials that aren't part of your final product will have an effect on the safety and often the efficacy of your product. One of the challenges, though, in terms of trying to understand how to qualify the safety risks of those products is that they're not directly regulated. And so a lot is left up to you as the manufacturer to figure out what is the acceptable margin of safety for any of my products. The guidances tell you how to think about that, but they leave the decision points open pretty broadly. So for us, with our supplement as a growth supplement for cell culture media, there are a few different alternatives that you have to think through. It's pretty much ubiquitous. Cell culture obviously needs some sort of growth supplement to keep expanding. And there's a range of things that are commonly used. FBS, obviously, as the workhorse, the standard for decades, moving more and more towards chemically defined media if it can be done, but that's clearly difficult and not always successful. And then the human-derived products, as Alex mentioned, AB serum and human platelet lysate are pretty broadly used these days. On the platelet lysate side especially, you have a choice between commercially available, ready-to-use platelet lysate or homebrew in lab preparation. I think if you look at the current changes and the opportunity, the pandemic and the unexpected distribution infection rates globally, the in-lab preparation becomes a little bit questionable in terms of, are you actually going to be able to maintain your supply on a daily basis when social distancing comes in? So then if you move to the commercial supplier, how do you evaluate that supplier and that product to make sure it's going to meet your safety needs going forward? So obviously our solution there, the human platelet lysate, is what we bring to the offer. And if you break down the way to qualify that and think about it from the performance, the safety, which is the quality and regulatory, and then the supply needs. So we have a range of platelet lysate products, and we say it's range because we think of this more as a product category than a single reagent. We started with Stimulate as a pooled human platelet lysate product with large donor batches, approximately 100 units per batch gives you a larger pool of material, quite limited variability across product. Moving then on to the pathogen reduction technology, as Alex mentioned, with Zika, raising questions about how do you ensure safety in the environment where you're not sure what might be coming along. Regulatory burden increases over the last few years in Europe and Japan, moving to expect pathogen reduction in some of these ancillary materials. And then now we're starting to evolve even further so that we have cell line specific products, such as our new T-Liven product, which is released against T cell growth verification instead of just broad general cell expansion. So on the safety side, the ancillary materials, as I mentioned, the guidances and sort of the regulatory expectations are reasonably established that you need to make sure your supplier has a robust supply of raw materials, that they're utilizing appropriately screened and tested donors, limit the additives or at least know what additives they have in there. And that gets you to that basic point. And most groups, and we certainly use FDA, AABB registered donation sites. We make sure that the platelet raw material was licensed as a transfusible unit, which ensures that you get that baseline safety aspect on there. The other thing that you can consider in a supplier is how many robust donation sites do they have? Do they have duplication? We have sites that are scattered throughout the country, allowing you to move supply from one place to another if there happens to be an outbreak in a region that is causing difficulty in collecting units. The quality is the same kind of aspects there, pretty typical quality system expectations. But as Alex mentioned, the ability to change what you're doing based on ongoing regulatory growth and changes is really important. The European Pharmacopeal expectations for pathogen reduction and risk assessment on virus really required everybody to sort of think about how are they going to do that with platelet lysate. And if you have a group that is not able to make those kinds of modifications, you're going to run into supply chain problems down the line. And of course, performance. Your material needs to be able to meet the expectations for your cell type And we really encourage people to consider that as it's a product category, the different preparation methods could have a significant impact, not just on cell number, but also the phenotype and how it performs in your final product. Another aspect of trying to improve the security and safety of the platelet lysate, the human-derived material supply, is in the manufacturing processes themselves. So clearly you need to have a GMP quality system to lock that down, but there are other points where you can improve the supply of the material and limit the risk from a supply chain perspective. We use, as do others, expired, outdated platelet units, as I mentioned before, that were previously intended to be utilized as transfusible units. We collect those from a range of different suppliers so that we have, as I said, backup on that, and we have a large inventory of raw materials that allows us to hold platelets for an extended period of time before we introduce them into our manufacturing chain. Along with that, making sure that you have validated traceability of your raw materials helps ensure that you can track back to any units that were donated after a period of time where an unexpected infection turned up. In our case, we're able to continue manufacturing with units for quite some time that were donated prior to the outbreak of COVID-19 in this case. Other typical standard safety measures that we have on there, limiting animal-derived materials, make sure you don't have any unknown contamination from there, and then just clean, good GMP manufacturing processes. So we mentioned the pathogen reduction technology that we introduced into our product, and talk a little bit about how we went about that process. So if you look into guidelines for finished product, viral inactivation and removal is pretty well discussed in guidance, ICH documents, the WHO technical reports, FDA expectations, and you can break down pathogen inactivation into two general methods. You have an inactivation process or a removal process. For us, we started looking at inactivation as that's pretty common throughout the FBS world. Some sera has gamma irradiated products. So we started a full robust development project to figure out what would be the best way for us to introduce a pathogen reduction technology without negatively impacting the performance of our product. So we did an analysis of several different methods, heat, chemical methods, radiation, a couple of different radiation methods. And we recognized that, as a complex biologic, it's very difficult to know exactly what you should be protecting against. And since we already have a product that has very limited additives to it, we didn't think it made any sense to add new chemicals or compounds into our products so that you would then have to characterize the risk of that. So sensitization agents for radiation didn't seem to make sense to us. Chemical removal didn't make sense. So we really honed in on just using straight irradiation. So both E-beam and gamma irradiation are pretty common methods to reduce the infectivity of pathogens in a product. We evaluated gamma initially because it is such a commonly used method of inactivation. And we found that just direct gamma irradiation of HPL, in our case, really affected the performance of the product and reduced the ability to support cell expansion. A lot of times you can add stabilizers to reduce the impact on off-target proteins, but as I mentioned, we definitely wanted to avoid any additives in our product. So we decided to move forward with a development project for E-beam. And E-beam, as you see, it has a very low penetration rate. And so we had to come up with a proprietary method to allow E-beam to impact the entire volume of our lots. We were successful with that. So once we developed that process, typical validation requires you to spike the product with representative viruses. The guidelines from ICH, viral risk assessment, want you to consider a range of different virus types so that you cover both RNA and DNA, enveloped and non-enveloped, large and small viruses. So with a pretty standard panel of representative viruses in our experiment, we went through and found that we were able to get an effective amount of reduction of virus in stimulate, now enliven and T-liven. The reduction range, if you are familiar with the expectations in finished products, Log reduction in the order of 1 is considered not effective. In the range of 2 to 4 is considered moderate to high effectiveness, and above 4 is sort of a highly effective reduction process. So we were pretty satisfied with the results of the irradiation process. And by doing it this way, we were able to end up with a product that had the same amount of growth support as the non-irradiated product without a very significant change. What we weren't expecting was that by irradiating the product, it ended up with some very interesting impact on the performance within certain cell types. And so just trying to think through what considerations you might want to review in thinking about how to address ensuring safety in a time of unknown outbreaks. Obviously, with any ancillary material, you start with, is it fit for purpose? But then making sure that your suppliers have a robust supply chain that can be modified in case of problems with any given region, looking at, do they have something to account for any force majeure or the robust quality product, and have they mitigated the safety risk appropriately? And then finally, Alex mentioned a little bit before the ability to adjust and the ability to respond. And we think it's very important to also make sure that your suppliers for all of your reagents understand your needs and can react to what's happening. And with that, we can move back to Dusty.
Thank you so much, Sean. Really appreciate it. So next up, we have Norihiro Watanabe from Baylor College of Medicine. Hiro, if you want to go ahead and take it away whenever you're ready.
Okay, can you hear me?
Yes, indeed. Thank you, Hiro. You're good to go.
Okay. So my name is Hiro. So I will be talking about like scientific aspect of the human platelet lysate, how HPL affect CAR T cell function if you use HPL during the ex vivo CAR T cell expansion. So far, as you might know, a number of clinical trials utilizing CAR T cells, especially targeting B cell malignancies, has been done. And from those clinical results, we have learned that good clinical response correlate to the longer T cell persistence or longer CAR T cell persistence in the patient. So one of the factors which contribute to the T cell persistence is a memory phenotype in the CAR T cell product as shown in this slide. So there's a fourth subset in the both CD4 and CD8 T cell. And we know the naive and central memory T cell, which can be identified by these surface marker expression, has a higher proliferative capacity and persistence once infused back to the patient. So in the ex vivo, during the ex vivo CAR T cell expansion, we want to maintain these two fractions as much as possible. So this is a basic like CAR T cell manufacture. We draw blood and stimulate PBMC and put cytokine to support cell growth and do CAR transduction, either lenti or retrovirus. And after around a week expansion, once you get enough number, we freeze CAR T cell product, and now it's ready for the infusion. So this is one of the example how CD8 memory phenotype looks like. And in this case, you can see like around 70% of less differentiated T cell phenotype, which is naive and central memory population. But after during this manufacture process, because of the stimulation or cytokine or CAR expression itself, in the end product, you lost most of the naive and central memory T cell. So one effort is focusing one effort is focusing to maintain these two fractions. by utilizing different method, which has been published before. But most of the case, they put additional factor which make GMP manufacture more complex and also increase cost. And importantly, during this manufacture process, most of the case, we use serum containing media. And so far, there's no comparison assay showing which serum affect, how serum affect CAR T cell function. So therefore, the aim of the study is to investigate the effect of different serum on CAR T cell expansion and CAR T cell function. So this is our experimental design. We take blood from healthy donor and stimulate with antibody and put cytokine, and we use retroviral transduction to express CAR. In this study, we use two different CAR. One is targeting prostate stem cell antigen, that's for the solid tumor, and the other one is targeting CD19 for B-cell malignancies. Since we wanted to see the effect of different sera during the T-cell expansion phase, we first generate CAR T-cell in the FBS, then we split into three different conditions, which is either FBS or human serum. And these tools have been used for the clinic for the CAR T-cell manufacture. And also we incorporate the HPL. And this has been used for the MSC, mesenchymal stem cell or stroma cell for the clinic. And after the around a week expansion, we performed a series of in vitro and in vivo experiments. So first we looked at the T-cell expansion. We have non-transduced unmodified T-cell and PSA and CD19 CAR. And in any case, we didn't see any huge difference in terms of the cell number. Then let's look at the memory phenotype. Once again, we are focusing on less differentiated T-cell phenotype. So when you culture non-transduced or expand non-transduced T-cell in the FBS, in this particular donor, you can get around 40% of less differentiated T-cell. But in the FBS, the percentage is reduced. However, if you use HPL for the expansion, you can get more or less differentiated T-cell, and this is a summary for the multiple donor. And this effect was also observed in the CAR PSA and CAR CD19. So in any case, we can enrich or maintain the less differentiated T-cell. So next we look at the function. So for the functional assay, we collect CAR T-cell expanded in different sera and wash out and set up in vitro experiment using FBS media to normalize the condition. So here I'm showing the short-term cytotoxicity assay, which is 4 to 5 hours assay. And in this case, when you use CAR PSA against PSA expressing cancer cell line, if you culture CAR T-cell in the HPL, their cytotoxicity is less than the other two. And this was more obvious when you use CD19 CAR. So it doesn't look good to use HPL for CAR T-cell expansion. But if you remember, we are enriching less differentiated T-cell, which has less cytotoxicity. Now you might be wondering, how about the proliferation then? So we perform proliferation assay. So in this case, we labeled CAR T-cell with specific dye and stimulate with tumor cell. And in this particular donor, if you see the histogram or this mountain shifted to the left, which means there are more proliferation. And in this particular case, and also multiple donor experiment, we demonstrated the HPL expanded CAR T-cell proliferate more upon antigen stimulation. We also checked T-cell death after this 5 days stimulation. And we observed there's less T cell death when you culture, when you expand CAR T cell in the HPL. And this was also seen in the CD19 CAR stimulated with NALM6 B cell leukemia cell line. There is more proliferation and less cell death in the condition which HPL cultured CAR T cell. So now, overall anti-tumor effect is a combination of those two, cytotoxicity and proliferation, and also with persistence of T cell. So to evaluate the overall function, we performed in vivo experiments. So this is showing the result for PSCA. We implanted PSA expressing tumor cell at the right flank, and 3 weeks later, we infused T cell, in this case labeled with GFP5 eluciferase, to track T cell expansion and migration. Now we are looking at T cell expansion from the bioluminescence imaging. So in contrast to in vitro experiment, in terms of T cell expansion at tumor site, we could not see huge difference, but in terms of anti-tumor effect, there's a huge difference. So this is a FBS treated group. So in this group, we actually achieved 1 mice which had complete tumor elimination, but all of them eventually relapse. Same for ABC-RAM. We could achieve 2 complete response, but relapse. But in the HPL condition, we achieved like 9 mice out of 12 for the complete tumor elimination, and 3 mice were still tumor free even after 100 days treatment. But as you can see here, this group also has tumor relapse. However, when we looked at the CAR antigen expression, in this case PSCA, in the FBS-ABS group, most of the relapsed tumor still express PSCA antigen. But in the HPL condition, only 3 express PSCA, and most of them are PSCA negative. So we believe CAR T cell did their job, but because of the antigen escape, we could observe the tumor relapse, which is out of our focus in this study. And this also translated into the better survival of the mice treated with HPL expanded CAR T cell. So since we couldn't see the difference in terms of CAR T cell expansion, now we want to focus on the T cell expansion and persistence. So we set up another experiment, which is very similar, but infuse more CAR T cells. And in this condition, in any condition, any CAR T cell, they can completely eliminate tumor. Then we decided to re-challenge new tumor on the left flank. Then we evaluated if the residual T cell could attack newly implanted tumor. So this is the bioluminescence imaging from T cell from the ventral side of the mice. So we are looking at T cell distribution and persistence on the day of the re-challenge. As you can see here, you can see like HPL treated, HPL expanded CAR T cells still stick around in the mice, especially in the lymph node or bone marrow. And this is a much promise summary. Once again, we are looking at T cell migration or expansion, in this case at newly tumor site, which is left flank. Only the HPL CAR T cell treated group, after we engrafted or re-challenged new tumor, you can see nice T cell accumulation at the newly tumor site and expansion, but not in the FBS and ABS treated group. In this graph, top showing the CAR T cell expansion at new tumor site. And as you can see here, HPL expanded CAR T cell expand at the new tumor site, and actually which eliminate or prevent the engraftment of the new tumor 4 out of 6 either delay tumor growth or completely tumor elimination. For the sake of time, I'm going to briefly show the CD19 CAR in vivo result. So top is anti-tumor effect. You can see in the FBS ABCM group, you can see tumor relapse at early time point, but HPL group can control tumor growth. And in this case, we could see the T-cell number difference between the group, either on day 8 and day 13. You can see high T-cell number in the peripheral blood from the mice. And this translated to the better survival to the HPL CAR T-cell treated group. So now interim summary, expanding CAR T-cell in the HPL media support maintenance of naive and central memory phenotype, which resulting in the potent in vivo anti-tumor response. So now we want to utilize HPL into a clinic. So the result I have shown so far is utilizing a healthy donor. But when we translate into the clinic, most of the case, we must use patient sample. So we first try to see if the HPL has similar effect, even if we use patient peripheral blood. So this is the patient PBMC who has B-cell lymphoma or B-cell leukemia and transduced with CD19 CAR. And if you look at the y-axis, unlike healthy donor, the fold expansion is very variable between the between the patient. But overall, there's no detrimental effect to use HPL media for the CAR T-cell expansion. So when we look at next we look at the phenotype after the around week 2 weeks expansion. So the gray is memory phenotype in the PBMC. Top is CD8 and the bottom is CD4. And as you can see, when you expand CAR T-cell in the HPL, you can maintain the percentage of less differentiated T-cell during the manufacture in both CD8, CD4. And from this result, we think we are not expanding naive and central memory T-cell specifically. We are actually maintaining their phenotype during the manufacture process. And then instead of performing the in vivo experiment, we performed 9 days in vitro co-culture experiment to see the cytotoxicity and T-cell proliferation. And the target cell is NALM6. And as you can see here, you can see the HPL expanded CAR T-cell showed nice anti-tumor effect compared to the other, and also showed a nice T-cell expansion. Okay, so the previous experiment so far, we use a little bit weird condition where we cultured, generated CAR T-cell in the FBS and they switched to the different media, 10% different media. So we, as an end user of the HPL, we of course want to reduce the cost first. So we ask if we can use lower percentage of HPL because there's no study using HPL for CAR T-cell expansion. And also to simplify the manufacture process, we ask, can we use HPL from the beginning? So to answer first question, now we use a range of serum concentration for the CAR T-cell expansion. So in this case, we haven't done any functional tests, but we checked the expansion and the phenotype. So as you can see here, CAR T-cells were able to expand even the 2.5% serum of the HPL and at similar level with 10% serum, but not in the 2.5% FBS and slightly lower in the AD serum. In terms of the phenotype, even the 2.5% HPL, you can still maintain higher percentage of less differentiated T cell, but this percentage is slightly lower than 10% or 5%. So I believe the 5% is kind of good deal to use in the GMP. So the next question is, to simplify the manufacture process, can we use HPL from the beginning? And again, we looked at, in this case, transduction efficiency and cell number and the phenotype. So this shows the CAR expression cultured in different media. So there's no detrimental effect for the CAR transduction efficiency. We observed the similar CAR expression in different media. And this is showing the fold expansion start from the beginning. And this 3 line is the original condition where we switch the media FBS to FBS or IB serum HPL. And when you use HPL from the beginning, you can see slightly higher number of the T cell expansion, but since the error bar is too huge, there's no statistical difference. And interestingly, if you use HPL from the beginning, you can get slightly higher percentage of CD4. And this was actually a good sign because there's a lot of literature showing 50% CD8, 50% CD4 CAR T cell mix can maximize the CAR T cell, overall CAR T cell function. So this could be a benefit. And of course, in terms of memory phenotype, you can maintain higher percentage of less differentiated T cell. So I just want to point out one thing. So if you use HPL from the beginning, at the end of the end product, we could observe, depending on the donor, we could observe NK cell expansion or contamination, although it's still less than 10%. And this is, I think, because we are using IL-2 for this culture, which support also NK expansion. But using IL-2 is a kind of old method. And now most of people utilize other cytokine like IL-715 cocktail. And when we use IL-715, there was no NK cell expansion. So in conclusion, as I shown, HPL can maintain less differentiated T cell, which promote overall, or which enhance overall anti-tumor effect. And HPL can be used at a lower percentage and also used as a complete replacement of the FBS and AD serum. And I didn't show the result, but when we use 5% HPL from the beginning, the CAR T cell still can expand and maintain the less differentiated T cell phenotype. So you can use, I haven't tried 2.5%, but at least you can use 5% HPL from the beginning. We start, this is a collaboration work between Baylor College of Medicine and Cook Regentech. Now it's Sexton Biotechnologies. And I'd like to thank all people which contribute to this study. Okay, that's all. That's it. Hello?
Thank you, Hiro. We got you. We're going to move on to some questions now that we have from our audience. Give us 1 second. The first question that we have is, does the COVID pandemic indeed lead to decreased availability of expired platelet concentrates? Sean, would you like to start by taking that?
Yeah, so this is actually a really interesting phenomenon that when a couple of weeks ago, you may have heard, certainly in the United States, there were reports of reduced blood donations, and in the UK, the same thing. And so we were trying to figure out, was that going to be an issue in terms of the supply? And it was an interesting thing happened where the Red Cross here in the United States and others put out a call for donations. And so as soon as there was a reduction, a lot of people actually went out and started to donate again. But what was happening was, given that a lot of places stopped doing a lot of optional procedures or elective procedures, and in the United States especially, there was a significant reduction in motor vehicle accidents, there ended up being a bit of a glut as soon as people started donating on platelets and blood supply. However, that's definitely dropped back off. And so there was an odd period about a week or two ago where there was a large amount of supply, but now that's starting to dry up and people are having some issues in terms of obtaining supplies.
Excellent. Thank you, Sean. Really appreciate that answer. Our second question, do you think the current COVID-19 issues will lead to changes in regulatory frameworks for blood-derived products? Alex, do you want to start us off on that one?
Yeah, I think I touched that a little bit in the presentation, where I do think that regulatory agencies will adopt new policies to address some concerns that have been raised by this current public health emergency, where it was difficult to quickly adopt new testing procedures after the pathogen had reached, that regulatory body's governing area, and they may be more aggressive in implementing new testing procedures prior to the first cases in for the FDA in the US or for the EMA in Europe, which is what we saw with Zika.
Excellent, thank you. Moving on to our next question. What if American blood banks implement pathogen inactivation on their platelet concentrates? Is the pathogen inactivation using E-beam as Sexton still necessary? Sean, that'd be directed at you.
I think that's open a little bit of an unknown at the moment. So the pathogen inactivation technology for blood products has been available in the United States and certainly in Europe for some time, and people had not picked it up, largely because the occurrence rate of contaminated products has been so low, and the extra expense just did not seem to really justify the level of risk mitigation. In the current climate, where we're thinking more often about frequent outbreaks of unexpected or unknown novel pandemics, it's certainly possible that people will migrate to just automatically doing some inactivation. If that happens, right off the bat, in terms of meeting the EP expectations of considering viral risk, you've got another layer of risk mitigation that's inherent in your starting material. But if you think about the way the ICH guidelines on viral risk reduction are laid out for manufacturing processes, they end up doing viral testing and viral risk reduction in large bioprocessing manufacturing, even though their starting materials have been extremely well characterized. So I think it's unclear how the regulators will respond to that, if that's going to be a sufficient amount of mitigation, or if they're going to still want some additional risk reduction.
Excellent. And I think we got time for maybe one more question. Give us 1 second here. Go with what is the lot-to-lot consistency of HPL?
So obviously, lot-to-lot testing and validation of every lot of material is a pretty common practice. And in FBS and AB serum, at least anecdotally, there's a lot of wide variability, and people are expecting that variability and test for it. In platelet lysate, we've really seen very low lot-to-lot variability. I mean, it's extremely consistent in terms of the growth ability with validated cell growth assays. We have customers that I think they all are experiencing essentially the same thing on that. We've looked at it not just from the growth perspective, but from biochemical analysis as well as protein concentration levels. And it's pretty striking how similar it is. I think we don't really exactly know why that is, but there's potentially a scientific explanation there, where platelet concentration and the proteins and everything that's expressed by platelets is really a genetic program, whereas the serum from any individual donor is going to be variable depending on what they ate or how recently they ate or what other conditions are affecting them. So can't say that for sure, but it's been strikingly consistent for platelet lysate.
Excellent. Thank you, Sean. And I think that's going to about wrap it up. I want to thank all of our panelists today, Alex, Sean, Hiro. Really appreciate the information. We'll be sending out both this webinar and a transcription of this webinar to our attendees. So we'll be on the lookout for that next week. And we hope everybody has a wonderful weekend. Thanks for tuning in.
Thank you, everyone.
Thank you, everyone.